So now we're going to look at replication. When DNA replication was first being studied, there were three major hypotheses as to how it was happening. One of them was the conservative model, one of them was the semi-conservative model, which we now know to be true, and the other one at the time was the dispersive model. So, we are going to look at the Meselson and Stahl experiment, and their hypothesis was that DNA replication happened semi-conservatively. So they grew a batch of bacteria in only N15, which is a heavy isotope of nitrogen, right here. Okay. And when this bacteria grew, they could only incorporate any type of heavy nitrogen when they were replicating. Because remember, the nitrogenous bases that help make up the nucleotides that build the DNA are going to, of course, incorporate nitrogen. So when they tested this sample, all the DNA was heavy, and that was what we can see right here at time 0. And when they took some of that bacteria in the N15 and they placed it in N14 media, that's now considered light. N14 nitrogen is light. And it grew then for several generations. So after 20 minutes, they went ahead and they tested the DNA. So again, at first you start with N15, right? And you can see this here. This is the N15. The other way that you can look at it is say both of these strands are heavy. They go ahead and they split. That's the heavy side. And then over here, they can only, when they're replicating, add light nitrogen to their molecules. So after 20 minutes, all the DNA was an intermediate. They were all basically heavy and light. And you can see that happening right here in generation 1, or time, the second time point where they're looking at 20 minutes. And the second generation then that happens is, again, you split off what you originally had. This would go here and that would go there. So all the new strands that you can add are, again, only going to be from the light N14 nitrogen. So as you start progressing through the generations, you see less and less of the heavy nitrogen and more and more and more of the light nitrogen being included. And that's going to go ahead and support this idea that DNA was replicating in a semi-conservative way. So what that means is when DNA replicates, the original template strands are going to go ahead and separate. So here are the parent molecules. They're going to go ahead and they're going to separate. Those are our templates. So here's a template and here is a template. And then the new nucleotides are going to go ahead and be built off of those templates, because if there's an A, you know T has to go ahead and be the base pair. If there's a C, you know that G has to be the base pair. And since this side is identical, what you end up having is two identical copies of the strand of DNA. And half of it is new. The light blue is the new. That's the semi part. And half of it is the old. That's the blue part. That's the conservative. And that's where you get semi-conservative from. So now what we're going to do is we're going to look at replication in a little bit more detail. So step 1 is going to be initiation of the replication bubble. So in order to replicate a genome of our size that is so large, you're going to have several replication bubbles occurring on the same chromosome at the same time so that you're speeding everything up. So in this process, you're going to have to know several enzymes, and one of these is going to be helicase. And what helicase does is it's going to go ahead and begin to unwind the DNA. And it's going to do that and clip the hydrogen bonds apart. And you can see the hydrogen bonds here breaking with that helicase enzyme. And then there's going to be these single-stranded binding proteins. Sometimes you'll see them as SSBP, single-stranded binding proteins. And they're going to bind to several of the nucleotides that have been split apart. And that's just to ensure that the DNA molecule doesn't reanneal or try to kind of stick back to itself. So because of the nature of the double helical structure of DNA, that massive unwinding that the helicase is doing is going to cause a problem for the head of the replication fork. It starts basically putting stress on the DNA. So topoisomerase is an enzyme that comes in and it clips a single base pair interaction. Okay, and it's only a temporary split because, hydrogen bonds can reform naturally, and this is just to alleviate the supercoiling that happens at the head of a bubble. Then there is primase. That's an enzyme that comes in and lays down specifically RNA, nucleotide bases, and they lay down what we call the RNA primer. And it's the primer that signals the DNA polymerase 3 to come and bind to that template strand to start adding new nucleotides. So that's where we have elongation. That's going to be step 2. That's where that DNA polymerase 3 is going to go ahead and begin to read that template. So it's going to read the template strand, and it's going to go ahead and lay down the complementary base pairs. So if it reads an A, it lays down a T. If it reads a T, it lays down an A. If it reads a G, it lays down a C. And if it reads a C, it lays down a G. So the DNA polymerase is an enzyme, which means it's very specific to what it can and cannot do. So this enzyme can only read when a primer tells it where to start. So in this case, you see a primer right there. Let me change the color so you can see a little better. You can see the primer right here. Okay, so that's signaling to this DNA polymerase, you can start here. However, it can only read the original template strand from 3' to 5'. It can only read 3 to 5. And you have to remember that DNA is built anti-parallel, which means that enzyme, if it's reading 3 to 5, has to lay down the base pairs 5' to 3. Okay? That means that the DNA polymerase 3 can only add nucleotides right here. It can only add this new nucleotide to a 3' hydroxyl end. So that's only looking at one side of the molecule. We're going to go ahead and go to the next slide and look at this in a little bit more detail. So this is going to pose a really interesting problem for the replication fork, because on the one side, the pole 3 is moving forward. So again, over here you've got your original 3', and this would be your original 5' template. You've got your primer, so your pole 3 binds here, and it starts laying down 5 to 3. And what that's doing is, as the helicase is here and unwinding in this direction, this strand is just going to go ahead and follow it. Okay? It's just going to follow behind and lay down the base pairs without an issue. And we call this strand the leading strand. But the other side of the DNA, this blue side, remember, it's still anti-parallel. So over here, you've got 3' and you've got 5' here. So that means that the helicase is moving in the opposite direction from how the DNA polymerase 3 can read. And that means that the enzyme has to do this in chunks. So this is the strand that we call the lagging strand. Here, the primase has to lay down the primer, and in this case, little primers are green, and it still lets the polymerase 3 know where to start. And it still reads the DNA template 3 to 5, but again, it's doing this in chunks. So for example, it started here and read forward, and then the helicase is progressing. It started here, it moved forward, but the helicase progressed. So now you have this open spot, and what needs to happen is another primer needs to come in here and sit down right there. So the primase will lay down the primer so that the polymerase can go ahead and bind here. And again, it's going to read 3 to 5, so it'll read in that direction and stop. All the while, that helicase is continuing to open up a spot. So these chunks that the lagging strand are going to be built in are called Okazaki fragments. In the end, the DNA is going to be replicated, but you can't leave the RNA fragments from the primer, okay? You can't leave these fragments in the DNA. You have to remove them. So that's done by an enzyme that we call DNA polymerase 1. This clips out the RNA nucleotides and replaces them with the correct matching DNA nucleotides. So you go ahead and you end up just fixing that. And then the ligase is going to go ahead and come in here, and any gaps to those Okazaki fragments, it's going to just literally ligate, okay? DNA ligase is going to come in here, and after the POL1 puts down the correct DNA base pairs, the ligase will come in here and take any of these breaks in the phosphodiester bond and smooth them over. It's going to ligate them. And then, of course, you're going to have proofreading enzymes like nuclease that'll go ahead and check the work. In eukaryotic chromosomes, the ends of chromosomes are going to be called telomeres. Okay, so the ends of the strands are going to be called telomeres. So telomeres don't contain genes, but they contain special nucleotide sequences that are made up of repeating sequences of a lot of T's and G rich. And since they're not made up of coding sequences, these telomere regions act like buffer zones when the chromosomes get shortened over many divisions. And it's been proposed that the shortening of the telomeres for each replication is maybe somehow connected to the aging process, or maybe that it's protecting against some cancer by limiting the number of divisions that some somatic cells can undergo. So the problem is, what about germ cells? What about certain stem cells that have to overcome this because they're dividing all the time? So the body has evolved a way to solve this replication problem on the ends of the chromosomes, again, for only these specialized cells. These are going to use an enzyme called telomerase. And telomerase is an enzyme that recognizes the tips of the chromosome. Okay, so it's going to recognize the tips of the chromosome, and it's going to recognize that it's missing a replicated section. And what that telomerase is going to do is it's going to go ahead and extend the parental piece of DNA so that there is room for an RNA primase to go ahead and lay down an RNA primer, so that there is a site where the polymerase can go ahead and bind and then add its sequence. And in this case, it's going to go ahead and extend that site so the polymerase 3 can bind and replicate that section. And that means that all the important genetic information is copied into our linear DNA. So that's not really a problem in the leading strand. It's really only a problem in the lagging strand. So now, when we're looking here, this is the last section. In bacteria, replication is slightly different. Okay, remember they only have one chromosome. Their DNA is, of course, double-stranded. There's still two strands. Some people get that wrong. It's still helical, okay? But the difference is it's circular, not linear like ours. So the replication is still semi-conservative. It's still bidirectional, but there's only one replication bubble. That means there's only one origin of replication. And in eukaryotes, that's what we've been doing the whole time. We mentioned again that there are multiple replication bubbles, and that's to again speed up the process. So that's replication. Hope that is helpful.